know about vomiting first is that it is protective. It's a reflex that helps to ward off unwanted substances that may have gained entry either through the eyesight or physical presence in the gastrointestinal system. So it has the different components. So now, being protective is a reflex. And the second thing you want to know is what is the vomiting center? What center controls vomiting? And that is the medulla. So you need to know that the medulla is the. Then what are the steps? Nausea first, retching second, and vomiting proper. The three steps in vomiting reflex is nausea, followed by retching, and then vomiting proper, or what you might call expulsion of substances through the oral cavity. Now, those are the basic. So in nausea, you feel like, uncomfortable with the situation. And then in retching, you find the abdominal muscle contracting, you find some form of, antiperistaltic movement, but the vomiting proper does not take place. So the end point of it is the forceful expulsion of substances out of the mouth. So, but how does it play out? There is this afferent and efferent components, which means the vomiting center has to receive sensation, has to be excited, and it processes the information and gives out an efferent output. This efferent output goes to a couple of muscles, a couple of sphincters that will contract powerfully, simultaneously, and then leading to expulsion of intestinal contents, or most importantly, stomach contents through the oral cavity. And if you have understood the physiology of the digestive system, you will now begin to visualize what is going to happen. So something cannot come out of the mouth if the lower esophageal sphincter is not open. The same thing with the cranial esophageal sphincter. There cannot be antiperistalsis without powerful contraction of the stomach and intestine in an antiperistaltic manner. There cannot be vomiting without a closure, normal vomiting without a closure of the epiglottis, without the epiglottis closing the larynx. Otherwise, there's going to be what we call aspiration pneumonia. The content is going to go into the trachea. But being a protective reflex and it's automatic, it is so coordinated that the epiglottis is going to do what? Close. During vomiting, respiration is a bit inhibited. So the diaphragm is going to be affected. The stomach contraction, the abdominal muscle, all of these will contract, all of those will contract at the same time. So that is one aspect. The other aspect is that you would have seen some substances that make you vomit without necessarily touching them. Sight. We're going to talk about the GIT. The GIT, we're going to be talking about the CTZ, that is the toxins that are found in the blood. We're going to be talking about vestibular stimulation, vestibular apparatus stimulation, vestibular stimulation. So all of this send sensory input to the medulla, to the vomiting center. So we're going to look at it this way. Now, the medulla is here. So for the eyes, the GIT, it will send physical, it will send information to that. Then the ear, the inner ear, the vestibular apparatus can also send information that. And then the next one is the CTZ. This is called the chemoreceptor trigger zone. Chemoreceptor trigger zone is a zone very close to the ventricles. Very close to the ventricles of the brain. And it is separated from the blood-brain barrier. So, it is very close to the blood-brain barrier. So solutes, some solutes, that area is very permeable and the blood-brain barrier is very strong. So this place does not have a strong blood-brain barrier. What is a blood-brain barrier? Blood-brain barrier is a tight junction created by astrocytes attaching to blood capillaries, which significantly prevents the movement of solids from blood into the brain. So that is very good to regulate increased osmolality of the brain cells, of the brain interstitium. If solutes will leak from capillaries into the brain, it's going to be predisposed to cerebral edema. So fluid will now move from blood into the interstitium, which is not good. So the blood-brain barrier prevents the movement of solids from cerebral capillaries into the interstitium, so that it wouldn't increase the osmolality of the brain interstitium. But these areas, the chemoreceptor trigger zone does not have sufficient blood-brain barrier, which means when blood comes to this area, certain chemicals in blood can seep out and excite this zone. And this zone also sends. So when food or toxins or whatever, or chemicals or drugs, whatever that is not very pleasant is found in the GIT. It could also be toxins from microbes, whatever is found in the GIT. There are chemoreceptors in the GIT that will send information to the medulla oblongata, which is the imaging center. By looking at nauseating stuff, then from the sensory input from the eyes goes to the medulla. Then the vestibular apparatus, during motion, the function of this vestibular apparatus is to make sure that the animal maintains balance, both of the head and of the eyes. So while movement takes place. However, the sensory nerves also go from this area to the medulla. So in the event of motion, not in every individual, in the event of motion, these sensory nerves that go to the medulla can also be significantly stimulated. So, and again from the CTZ. So all of these make their afferent inputs. They make their afferent inputs to the medulla. And then when medulla receives this, it now sends out motor fibers to some muscles. One, the diaphragm. Two, the stomach. Three, the abdominal muscles. 4, lower esophageal sphincter. 5, cranial esophageal sphincter. 3, duodenum. Abdominal muscles. So these efferent inputs, so these are now efferent. These efferent inputs will go to all of this. This will relax. This will relax. This will contract. This will contract, this will contract. Then the epiglottis also closes the larynx at that time. So with the powerful contraction of those that with the coordinated activity of all of this, there's going to be forceful projection of intestinal or stomach contents through the oral cavity. Does that make sense? Good. So what are the takeaway points? One, know the central role of the medulla as the vomiting center. Know that vomiting is due to what? Sensory input from different areas of the body into the vomiting center. So, vomiting can result from any of this. CTZ, vestibular apparatus, gastrointestinal system, and sight. So when the medulla receives this sensory input, it will do what? It will now give out a motor control to all of those muscles. Some will relax, sphincters will relax, muscles will contract, and then there will be antiperistalsis. Okay. So in such cases, catecholamines, for example, what type of receptor is going to be activated here? H1 receptor, histamine 1 receptor. Histamine 1 receptor. Those chemicals that may be detected by the CTZ in the body acts on histamine 1 receptor, which means histamine is a good facilitator of what? Vomiting. So how do you control it? Antihistamines or anticatecholamines. And this group of catecholamines can be what? Dopamine, histamine, serotonin. So to manage this condition, you may want to use antiemetics. The movement of materials within the colon become delayed. This synergification. The third one is obstruction of the colon. So that's going to cause a problem called obstructive defecation. This may be caused by rectal prolapse, colibases on their own are enzyme. They help to prevent the inhibitory effect of bile salts on pancreatic lipase. So with that, the pancreatic lipase can easily act on the lipids, the triglycerides found in diet. And when the triglycerides have been broken down, it's broken down into fatty acids and glycerol. So fatty acid and glycerol will now be absorbed. They will now be absorbed into the enterocytes. When they are absorbed into the enterocytes, they will recombine, what we call re-esterification. They will recombine through an ester bond to form triglycerides again. So as triglycerides, it cannot be transported in blood. So it has to be attached to a protein called apoprotein C. So inside the enterocytes, a protein called apoprotein C unites with the triglyceride, and which are in smaller units though, not as large as it were as in the diet. When it now bonds with when the triglycerides, small molecules of triglycerides bind with the apoprotein C, it is now called what? A chylomicron. Until this happens, lipids will not be transported in the lacteals. So once it has entered, once it now has chylomicrons, it will now be absorbed directly into the lymphatic vessel. You remember we said there is lactates. The first thing is when carbohydrates are broken down as glucose molecules, the glucose enters the cells. When they enter the cell, the first thing that happens is glycolysis. When glucose enters cells, because of the low amount of oxygen in the cytoplasm, it will undergo glycolysis. So this glycolysis is anaerobic and essentially yields pyruvate. So glycolysis will now yield pyruvate. So this takes place in the cytoplasm. That's what happens to glucose. And then this pyruvate yields very small amount of energy, ATP. And then it produces pyruvate. Then pyruvate moves into the mitochondria. Two molecules of pyruvate produced. Then pyruvate moves into the mitochondria and undergoes decarboxylation reaction. It undergoes decarboxylation reaction inside the mitochondria. So it is imported across the inner mitochondrial membrane and it moves into the mitochondrial matrix, undergoes decarboxylation reaction, producing carbon dioxide and acetyl-CoA. Produces carbon dioxide and acetyl-CoA. Then acetyl-CoA, of course, other electrons are also put, high energy compounds which cannot be used by the TCA cycle. So acetyl-CoA will now enter into the TCA cycle, happening here. Tricarboxylic acid cycle happening here. So it first of all reacts with what? I'm not going to tell you the TCA cycle. It's going to give you oxaloacetate. First of all, converted to oxaloacetate and converted to citric acid or citrate. Then citrate will now enter that cycle and undergo up to 8 to 9 stages of reaction, from succinate to malate to isosuccinate and all of that. But okay, you can always go and check it and check that cycle. So acetyl-CoA will now enter into the TCA cycle, but it is going to first of all react with oxaloacetate and then to produce a citrate. So citrate or what we call citric acid, citrate or citric acid will now enter, will be the precursor for that cycle, and then it will move. It will have up to 9 stages. So this is the general pattern. So now it's the 9 biochemical steps make up. This is hydrogen. These are hydrogen carrying molecules. So hydrogen in the mitochondria, they will all be transported into the inner mitochondrial matrix. As this hydrogen is being transported into this place, their concentration here increases and their concentration in the mitochondria decreases. So there is a gradient, significant gradient for what? For electrons. So electron carriers, cytochromes, and there are different types of cytochromes. Cytochromes will move this electron produced by the Krebs cycle within the mitochondrial matrix away from the mitochondrial matrix and import them here, creating a significant amount of, a significant gradient for what? For electrons being high in the inner mitochondrial membrane. what happens to a substance when it is in high concentration in a particular compartment compared to the other? It's going to diffuse back. So this hydrogen that is now high in the inner mitochondrial membrane will tend to do what? Diffuse back. That diffusion activates an enzyme called ATP synthase. ATP synthase. So once the proton begins to diffuse back, this enzyme is activated and it catalyzes the addition of ADP plus phosphate to produce ATP. Interestingly, this produces the largest amount of ATP. 22 molecules of ATP is produced. So in the end, glycolysis produces how many? 2. Krebs cycle produces how many? 14. And then